Our next lesson is on designing studies using experiments. In this section, we'll look at observational studies from experiments, describing the language of experiments, applying 3 principles of experimental design, and looking at design comparative experiments using completely randomized designs and randomized block designs, including matched pairs design. Experiments include some treatment in order to measure a response. An observational study observes individuals and measures variables of interest but does not attempt to influence the responses, whereas an experiment deliberately imposes some treatment on individuals to measure their responses. When our goal is to understand cause and effect, experiments are the only source of fully convincing data. The distinction between observational study and experiment is one of the most important in statistics. The November 2009 issue of Nutrition Action discusses what the current research tells us about the supposed benefits of soy. For a long time, scientists have believed that the soy foods in Asian diets explains the lower rates of types of cancer and heart disease. However, when the experiments were conducted, soy either had no effect or a small effect on the health of the participants. For example, several different studies randomly assigned elderly women either to soy or placebo, and none of the studies showed soy was more beneficial for preventing osteoporosis. So what explains the lower rates of osteoporosis? We still don't know. It could be due to genetics, other dietary factors, or other differences between Asian cultures. Observational studies of the effect of one variable on another often fail because of confounding variables between the explanatory variable and one or more lurking variables. A lurking variable is a variable that is not among the explanatory or response variables in a study, but that may influence the response variable. Confounding occurs when two variables are associated in such a way the effects on a response variable cannot be distinguished from each other. Well-designed experiments take steps to avoid confounding. Lurking variables create problems in 2 different ways. They can create extra variability in the response variable and also have the potential to become confounded when the explanatory variable is not controlled. Does reducing screen brightness increase battery life in laptop computers? To find out, researchers obtained 30 new laptops of the same brand. They chose 15 of the computers at random and adjusted their screens to the brightest setting. The other 15 laptop screens were left at the default setting, moderate brightness. Researchers then measured how long each machine's battery lasted. Was this an observational study or an experiment? Let's take a look carefully at the example. This would be classified as an experiment. Does eating dinner with your families on a sample survey conducted by researchers? This would be an example of an observational study, not an experiment. There was no experiment or anything actually imposed. The explanatory variable would be how many times a week the children are eating, and that would be for how many meals. That would be 5 times a week. And the response variable are most likely their GPAs. They're more likely to get better grades in school, so most likely this had to do with their GPA. Lurking variables, there can be many different types of lurking variables. For example, it could be just simply that students whose families were sitting down to eat with their families, their parents more interested in education, for example, than perhaps other families. An experiment is a statistical study in which we actually do something, a treatment to people, animals, or objects, which are called the experimental units, to observe the response. Here's the basic vocabulary for experiments. A specific condition applied to individuals in an experiment is called a treatment. If an experiment has several explanatory variables, a treatment is a combination of specified values of these variables. The experimental units are the smallest collection of individuals to which treatments are applied. When the units are human beings, they're often called subjects. Sometimes explanatory variables in an experiment are called factors. Many experiments study the joint effects of several factors. In such an experiment, each treatment is formed by combining a special value of each of the factors. Often these are called levels. A study published in the New England Journal of Medicine compared two medicines to treat head lice: an oral medication called ivermectin, a topical lotion containing malathion. Researchers studied 812 people in 376 households in 7 areas around the world. Of the 185 randomly assigned to the ivermectin, 171 were free from head lice after 2 weeks, compared to only 151 of the 191 households randomly assigned to the malathion. We identified the experimental units, explanatory and response variables, and the treatments in this experiment. So in this example, the experimental units are the households, not the individual people, so that since the treatments were assigned to entire households, not separately to individuals within the households. So again, experimental units in this case are households. The explanatory variable is type of medication, and the response variable in this instance, or example, is whether the household was lice-free. The treatments in this instance were the 2 types of medicine. I have them circled here and I'll arrow down. Here's another example. Does adding fertilizer affect the productivity of tomato plants? How about the amount of water given to the plants? To answer these questions, a gardener plants 24 similar tomato plants in identical pots in his greenhouse. He will add fertilizer to the soil in half of the pots. Also, he will water 8 of the plants with 0.5 gallons of water per day, 8 of the plants with 1 gallon of water per day, and the remaining 8 plants with 1.5 gallons of water per day. At the end of 3 months, he will record the total weight of tomatoes produced on each plant. Identify the explanatory and response variables, the experimental units, and list all of the treatments. The 2 explanatory variables in this example are fertilizer and water. The response, so I'll circle the 2 things here and then just arrow down so it's a little easier for me to show that. The response variable is the weight of tomatoes produced. The experimental units are the tomato plants, and the treatments. There are 6 treatments. Fertilizer with 0.5 gallons of water, fertilizer with 1 gallon of water, and fertilizer with 1.5 gallons of water. The same thing for no fertilizer. No fertilizer with 0.5 gallons. There will be no fertilizer with 1 gallon, and no fertilizer with 1.5 gallons. So each of these, the 0.5 gallons, and the 1.5 gallons of water, will have treatments for fertilizer and no fertilizer. When there's only one factor or explanatory variable in an experiment, the levels of the factor are equivalent to the treatments. When there's more than one factor, the combinations of each level of factor form the treatments. Experiments are the preferred method for examining the effect of one variable on another. By imposing a specific treatment of interest and controlling other influences, we can pin down cause and effect. Good designs are essential for effective experiments, just as they are for sampling. A high school regularly offers a review course to prepare students for the SAT. This year, budget cuts will allow the school to offer only one online version of the course. Over the past 10 years, the average SAT score of students in the classroom course was 1,620. The online group gets an average score of 1,780. That's roughly 10% higher than the long-term average for those who took the classroom review course. Is the online course then supposedly more effective? It's hard to tell. Many laboratory experiments use a design like the one in the online SAT course example. We have experimental units, a treatment, and we measure a response. In the lab environment, simple designs often work well. Field experiments and experiments with animals or people deal with more variable conditions. Outside the lab, badly designed experiments often yield worthless results because of confounding. Does caffeine affect pulse rate? Many students regularly consume caffeine to help them stay alert. Thus, it seems plausible that taking caffeine might increase an individual's pulse rate. Is this true? One way to investigate this is to have volunteers measure their pulse rates. Drink some cola with caffeine, measure their pulses again after 10 minutes, and calculate the increase in pulse rate. Unfortunately, even if every student's pulse rate went up, we couldn't attribute the increase to caffeine. So perhaps it was sugar in the cola or some other reason of why this occurred. So these are just some examples that there are many variables that are potentially confounding when we use caffeine, for example, in our study. The remedy for confounding is to perform a comparative experiment in which some units receive one treatment and similar units receive another. Most well-designed experiments compare one or two treatments. Comparison alone, though, isn't enough. If the treatments are given to groups that differ greatly, bias will result. The solution to the problem of bias is random assignment. In an experiment, random assignment means experimental units are assigned to treatments at random, that is, using some sort of chance process. In a completely randomized design, the treatments are assigned to all the experimental units completely by chance. Some experiments may include a control group that receives an inactive treatment or an existing baseline treatment. So we have our experimental units with random assignment, our two groups, treatments, and then compare the results. Suppose you have a class of 30 students who volunteered to be subjects in the caffeine experiment described earlier. How would you randomly assign 15 students to each of the two treatments? One way we could do this, for example, is to have 30 slips of paper, write A on 15 of them, write B on the other 15. Mix them in a hat and have each student select one. Have each student who received A drink the cola with caffeine, and each student who received B to drink the cola without caffeine. A healthy organization wants to know if a low-carb or low-fat diet is more effective for long-term weight loss. The organization decides to conduct an experiment to compare these two diet plans with a control group that is only provided with brochures about healthy eating. 90 volunteers agreed to participate in the study for 1 year. Here we want to outline a completely randomized design for this experiment. So again, what I would do for this one, for example, is to assign 3 groups. Group 1, which would have 30 subjects. Group 2 would have 30 subjects, and Group 3 which would also have 30 subjects. Group 1 for the treatment would have the low carb. Group 2 would have the low fat. And group 3 will be the control group. Then we compare weight loss in all three of these designs with these treatments. Here's another example. Music students often don't evaluate their own performances accurately. Can small group discussions help? The subjects were 29 students preparing for the end-of-semester performance, an important part of their grade. We assign 15 students to the treatment, videotape a practice performance, ask the student to evaluate it, then have the student discuss the tape with a small group of other students. The remaining 14 students form a control group who watch and evaluate their tapes alone. At the end of the semester, the discussion group students evaluated their final performance more accurately. Outline a completely randomized design for the experiment. So here's an example of how that would look. 50 volunteer students, random assignment, group 1, 25 students, group 2, 25 students. Treatment online, treatment 2 classroom, and then we compare the scores. Describe how we would carry out a random assignment for this experiment. So example, here's what it would look like. We would again use random assignment. We could again use different things, random generate from a calculator, pieces of paper, things like that to be able to perform the actual experiment. Randomized comparative experiments are designed to give good evidence that differences in the treatments actually cause the differences we see in the response. Principles of experimental design include control for lurking variables that might affect the response. Use random assignment, and use enough experimental units in each group so any differences in the effects of treatment can be distinguished from chance difference between the groups. How this would work with a placebo, and then using the different types of medicine. So a placebo is a dummy pill or an active treatment, indistinguishable from the real treatment. You'll see a placebo often used in experiments, especially experiments where they are trying to look at effects of different kinds of medicines. Here's our example back with the caffeine example. Explain how to use all three principles of experimental design in the caffeine experiment. I'll just explain how each of these would work. Control, first of all, we'll start with, and I'll go through again and explain. So our first one is control. There should be a control group that receives non-caffeinated cola. The subjects in each group should receive exactly the same amount of cola served at the same temperature. Also, each type of cola should look and taste exactly the same and have the same amount of sugar. Subjects should drink the cola at the same rate and wait the same amount of time before measuring the pulse rates. If all of these lurking variables are controlled, they will not be confounded with caffeine or be an additional source of variability in the pulse rates. The next thing we look at is randomization. Subjects should be randomly assigned to one of the two treatments. They should roughly balance out the effects of the lurking variables we cannot control, such as things like body size, caffeine tolerance, and the amount of food recently eaten. Our third item we'll look at is replication. We want to use as many subjects as possible to help make the treatment groups as equivalent as possible. This will give us a better chance to see the effects on caffeine or of caffeine if there are any. The logic of randomized comparative experiment depends on our ability to treat all the subjects the same in every way except for the actual treatments being compared. The logic of a randomized comparative experiment depends on our ability to treat all subjects the same way. Good experiments, therefore, require careful attention to details to ensure all subjects really are treated identically. A response to a dummy treatment is called the placebo effect. The strength of the placebo effect is a strong argument for randomized comparative experiments. Whenever possible, experiments with human subjects should be double-blind. In a double-blind experiment, neither the subjects nor those who interact with them and measure the response variable know which treatment a subject received. Here's just another example. Researchers discovered placebos have a stronger effect when they are perceived to be more expensive. The study had volunteers rate the pain of an electric shock before and after taking a new medication. However, half the subjects were told the medication cost $2.50 per dose, while the other half were told the medication cost 10 cents per dose. In reality, both medications were placebos, and both had a strong effect. So researchers suggested people are accustomed to paying more for better medications, which may account for the difference in response. And as with any placebo, it's all about the expectations of the subjects. In an experiment, researchers usually hope to see a difference in the responses so large it is unlikely to happen just because of chance variation. We can use the laws of probability, which describe chance behavior, to learn whether the treatment effects are larger than we would expect to see if only chance were operating. If they are, we call them statistically significant. So an observed effect so large it would rarely occur by chance is called statistically significant. Here's an example from your book where a simulation was completed about drivers and distracted drivers and whether or not people would stop or fail to stop for those who were using cell phones. We will take a look at this probably in class. Here's another example. This is an example using chocolate chip cookies. Again, this shows an example of how the design can be carried out in an experiment and how the diagram would look. We have chocolate chip cookies, 20 locations, top rack for 10, bottom rack for 10. Randomly assign the cookies to the locations, and then we have two types of two different recipes, that is, A and B. And then when we're done, we compare the taste. You can read through again the entire experimental design is listed on here, and then here's an example of how it would look. Completely randomized designs are the simplest statistical designs, but just as with sampling, there are times when the simplest method doesn't yield the most precise results. A block is a group of experimental units known before the experiment to be similar in some way that is expected to affect the response to the treatments. In a randomized block design, the random assignment of experimental units to treatments is carried out separately within each block. We form blocks based on the most important unavoidable sources of variability or lurking variables among the experimental units. Randomization will average out the effects of the remaining lurking variables and allow an unbiased comparison of the treatments. Control what you can, block on what you can't. Control and randomize to create comparable groups. For our better texting example, because the subjects include people of varying texting abilities, a completely randomized design would lead to lots of variability in the response variable. In a randomized block design, the experienced texters would be compared to each other, and the novice texters would be compared to each other. This will make it easier to see a difference in the response variable if there indeed is a response variable in the response in this example. To randomly assign the subjects to the two groups, we would put 50 slips of paper labeled A in a hat and 50 slips of paper labeled B in a hat. Mix up the papers, have each subject select one to determine which design he or she will use. We follow a similar process for the 200 novice texters. After the experiment, compare the mean number of words correct for the two different designs within each block. So we would start again with 300 subjects. We would have 100 of the experienced texters and 200 in the novice. We would then have each one do random assignment. This would be for both of them. We would then, in the first group, have 50 look at type A, and we would have 50 look at type B. We would then compare the mean number correct. We would do the same in the other, except for this one, we would still do a random assignment. We would do 100 for type A, and we would do 100 for type B. And again, when we're finished, we would compare the mean number correct. And that's it. I would write everything out if I were actually answering this question. Due to limited space on here, I'm not writing everything out. Matched pairs design is a randomized block experiment in which each block consists of a matching pair of similar experimental units. Chance is used to determine which unit in each pair gets each treatment. Sometimes a pair in a matched pair design consists of a single unit that receives both treatments. Since the order of the treatments can influence the response, chance is used to determine which treatment is applied first for each unit. Looking ahead, we'll look at studies and using studies wisely.